Hello, I'm Dang, and my research focused on the analysis of alternatively spliced gene in stickleback and how it affects the species to adapt to various environments. My research question is, does alternative splicing play a role in stickleback adaptation to different environments? Some background on my research is that we are looking at molecular changes via alternatively spliced gene of interest, genes that have been arranged differently through modification via a cellular level in stickleback to see how it affects environmental adaptation. Looking at alternatively spliced gene would help us figure out how the three-spine stickleback adapted to river and lake environments so quickly, since alternatively spliced gene play a role in differentiation and diversity. We discovered 274 genes with different pattern of alternatively splicing between population of stickleback that lives in various environments. To see if alternative splicing has a role in stickleback adaptation, we use PEAT, or pathway enrichment analysis. How PEAT works is that it takes in a list of genes and see what biological pathway is most present or enriched. And you can do this with a database or through coding. We use a pathway enrichment analysis because it would allow us to determine if different spliced genes between population of stickleback, river versus lake are enriched in certain biological pathway. This would allow us to see what pathway may be differentiated between population. An example of a database for pathway enrichment analysis was David Bioinformatics, which I used for my enrichment. With David Bioinformatics, all I had to do was plug in a list of gene of interest, select the species and type of ID I had on my list. For my summary results, since I'm viewing pathway enrichment, I would choose KEGG pathway. KEGG pathway is a biological pathway that is directly connected to the KEGG database. For my list of interests, the KEGG pathway that was enriched was Spliceosome pathway, highlighted in red. The pathway saw enrichment in the p-value and Fisher x-axis, but not for full enrichment, Bonferroni, Benjamini, and FDR. I also had 4 genes in the pathway that were enriched. These 4 genes all play crucial roles in spliceosome pathway, which include regulating intron removal in the pre-mRNA to mRNA, facilitating splicing through recruitment of more splicing protein, regulating splicing expression, and promoting or inhibiting splicing from happening. From our result, we hypothesized that alternative splicing itself is regulated through splicing. This would mean regulation in splicing would allow for stickleback to adapt to various environments. Since crucial genes involved in spliceosome pathway function were altered via splicing, this would lead to various ways population of stickleback splice their genome. Having differently spliced genome would increase genetic diversity and variation that allow for their ability to adapt to various environments. Thank you for listening to my presentation.